Cellular respiration is a series of reactions within the cytoplasm and mitochondria of eukaryotic cells in which glucose is broken down into energy to form ATP. This process happens in 4 steps: glycolysis, pyruvate oxidation, the Krebs cycle, and electron transport chain. This is the equation for cellular respiration. Glucose molecules have a backbone of 6 carbons, 12 hydrogen, and 6 oxygen. In a synthesis reaction, ADP and an inorganic phosphate create ATP, which is used as energy. Energy is released as Gibbs free energy, which creates ATP. It is created through many reactions in the cytoplasm and mitochondria. Glycolysis occurs in the cytoplasm, pyruvate oxidation and the Krebs cycle happen in the matrix, and electron transport chain occurs in the inner membrane space. Cells oxidize food without oxygen through anaerobic respiration or fermentation. There are 2 types of anaerobic respiration, the first being obligate, meaning it must not be in the presence of oxygen, the second being facultative, in which it can be with or without oxygen. There are 4 steps in cellular respiration: glycolysis, pyruvate oxidation, Krebs cycle, and electron transport chain. Glycolysis produces 2 ATP. Pyruvate oxidation does not create ATP, however it creates acetyl-CoA to be transferred to the Krebs cycle, where 2 more ATP are formed. In ETC, approximately 32 ATP are produced to finish off cellular respiration.
Glycolysis is the first stage of cellular respiration. ATP phosphorylates glucose into G6P. G6P becomes F6P through isomerization. ATP phosphorylates F6P into F16BP. F16BP splits into DHAP and G3P. DHAP converts to G3P, so end result is 2 G3P molecules. 2 G3P are converted to BPG and NAD+, gets reduced to NADH. BPG is converted to 3PG. A phosphate group from BPG phosphorylates ADP to ATP. 3PG is rearranged into 2PG. It becomes an isomer. 2PG is converted to PEP, or PEP, by removing water. PEP becomes pyruvate. A phosphate group leaves PEP and phosphorylates ADP into ATP. Pyruvate oxidation is the second stage of cellular respiration. For cellular respiration to continue, 2 pyruvate molecules must migrate from the cytoplasm to the mitochondria. The pyruvate molecules move into the mitochondrial matrix with the help from the membrane carrier. The pyruvate then passes through the outer and inner mitochondrial membranes. Coenzyme A latches onto the acetyl group of a pyruvate molecule. This allows coenzyme A to be oxidized and NAD+ to be reduced into NADH. Once this reaction is done, CO2 is removed as waste. This results with acetyl-CoA, which then moves to stage 3 of cellular respiration, the Krebs cycle. The Krebs cycle is the third stage of cellular respiration. The acetyl group from acetyl-CoA combines with oxaloacetate to make citrate. Citrate becomes isocitrate through isomerization. Isocitrate is oxidized to become alpha-ketoglutarate by removing CO2 and 2 hydrogens. NAD+ gets reduced to NADH. Alpha-ketoglutarate becomes succinyl-CoA through oxidation by losing CO2, and NAD+ becomes NADH through reduction. Succinyl-CoA becomes succinate through oxidation by releasing coenzyme A, and ATP is created through substrate phosphorylation. Succinate becomes fumarate through oxidation, and FAD becomes FADH2 through reduction. Fumarate is converted to malate. Malate is oxidized to oxaloacetate, and NAD+ is reduced to NADH. NADH and FADH2 are created through the first 3 stages of cellular respiration. In stage 4, electron transport chain, is where they are converted into ATP. NADH attaches to complex 1, and the hydrogen is removed, leaving NAD+. The hydrogen then travels to complex 3, 4, and finally ATP synthase, where that hydrogen carries enough energy to produce ATP from ADP. FADH2 attaches to complex 2, and the hydrogen is removed, leaving FAD. The hydrogen then travels to complex 3, 4, and finally ATP synthase, just like the hydrogen from NADH. The hydrogen then carries enough energy to produce ATP from ADP. This process is repeated until approximately 32 ATP are created. This concludes the 4 stages of cellular respiration.